Hello, guys. In this lecture, I would tell you that how the B lymphocytes or the B cells produce immunoglobulins or antibodies that are very diverse in nature. Uh, every B cell produces a unique type of immunoglobulin or antibody that is part of its membrane, or you can say the antibody or immunoglobulin is attached to the membrane of the B cell. We also call this immunoglobulin is the B cell receptor, or this antibody is called the B cell receptor. And every B cell is produced with a unique type of an antibody attached to its surface. So how this variability is ensured at the genetic level, because there can be numerous types of antigens like different types of bacteria, different types of viruses or other antigens that can enter into the body, and your immune system is going to respond against all those antigens. The adaptive immune system, particularly the important cells like the B cells, with the help of the T cells, produces a weapon against all those antigen, and that is the antibody. And that weapon is highly, specific, and it specifically targets, that specific antigen that has entered into the body. Now, there are two types of processes you should remember that happen here. Um, One is somatic recombination. This, you would have heard about this term before. And the second process that happens for this variability in the B cell receptor or antibodies, that is somatic hypermutation and class switch recombination. Now you should remember that the B cell, when it is undergoing its development, you know that all the blood cells, they are produced uh, in the primary lymphoid organ, which is the bone marrow. So inside the bone marrow, when the B cell development is taking place, there would happen this rearrangement of the genes that are responsible for coding these proteins, the immunoglobulin membrane immunoglobulins are the B cell receptors. So those genes that are responsible for making this antibody on the surface of the B cell um, actually are rearranged during this developmental process, and every B cell has to undergo this particular process of somatic recombination. So that is the first thing that should happen to each and every B cell. The second process that would happen to certain types of B cells, but not our B cells. What are those B cells? The second process, the name of the second process is somatic hypermutation or class switch recombination. It doesn't happen to all the B cells. It will happen to a B cell who has come in contact with an antigen, or the B cell which has become an effector B cells. You know that this antibody is expressed on each and every B cell, but a B cell that has come in contact with an antigen has found its perfect partner in the shape of a perfect antigen that binds to the B cell receptor. That particular B cell becomes an effector cell. It proliferates. Many clones of the same cells are made in the lymphatic system or inside the lymph nodes. In later, the B cells would become either a plasma cell or a memory cell. So the plasma B cell would secrete the same type of antibody. It would synthesize the same antibody and would not, make this antibody attached to its surface. It would rather secrete the same immunoglobulin antibody, which actually perfectly bound to that particular antigen. So uh, then inside that particular cell, or that particular B cell, which has become an effector cell, further fine tuning of the antibody molecule or the immunoglobulin is needed. And that is done with the help of this process, somatic hypermutation or class switch recombination. So the first thing that how this somatic recombination actually takes place in each and every B cell, and what are the underlying processes, how this recombination is happening there. So this is an antibody molecule that you see right here. And in this antibody molecule, there are some red boxes at the top, and there are some blue boxes that you see here. Every antibody has a light chain which is flanking this Y-shaped structure. This is the light chain on one side, and this is the light chain on another other side, and two heavy chains. One heavy chain is here, one heavy chain is here. So this is making a Y-shaped structure. In case of both the heavy chains and the light chains, these, red boxes that you see here at the top of this polypeptide or this immunoglobulin, there is the antigen binding site. So the antigen binds to these arms of the antibody that you see here and here on one side and another side. And this side uh, which usually in a B cell, which is not an effector cell, it is anchored into the membrane of the B cells with the help of a transmembrane domain. It has got an FC receptor. This region is called the FC region, or FC means fragment crystallizable. It means this is a crystallizable part of the antibody. So for this fragment, crystallizable fragment, the receptors usually are found on many immune cells, like the mast cells or the different types of phagocytes, like macrophages, etc. And this can, uh, help with the process of opsonization when it attaches to the immune cell, like a macrophage on one side and an antigen on another side. So this facilitates the process of phagocytosis. We call that opsonization. Now, uh, the variability should be ensured here at the antigen binding site, or the red boxes that you see here. The red boxes are for the variable regions that are found in the light chain as well as in the heavy chain. Both the light chain and heavy chains have these red variable regions. Now, if you look at the genetic level, the genes that are coding for these peptides, whether these are peptides of the light chain or peptides of the heavy chains, they are not located on a single chromosome. For instance, the light chain can be of two types in an antibody. It can either be a lambda light chain, or it can either be a kappa light chain, two types of light chain. In antibodies, you can have one type of a light chain and one type of an antibody, never both. So uh, this light chain, if it is a lambda light chain of an antibody, this is encoded by genes that are present on chromosome number 22. And this gene is not a single stretch of coding, DNA. This actually is found in segments, and these segments are dispersed on chromosome 22. For instance, these red boxes that you see here, these actually are different segments of the variable region of this particular antibody. So V lambda 1 means this is the first segment of the variable region of this lambda light chain locus. This segment is to be joined with this particular segment, and this segment is joined to be these segments. How many of variable segments could be found in the case of this lambda light chain? There could be up to 30 different, segments in that when combined would code for this particular variable region of this antibody. So 30 different segments are found on chromosome 22, and there is a lot of intervening DNA between these, segments. You can just imagine, and those intervening segments should be taken out of this DNA in order to bring them close together. So this is accomplished with the help of this process of recombination or somatic recombination, this gene rearrangement that takes place here. They are combined in different ways. So you would find an antibody that would have a variable region. One antibody would have one type of variable region, another would have different types of variable region, because these would be combined in different ways. There are different rules at the genetic level. Different enzymes are involved in this particular process. So it means that all red boxes should be, brought together in order to make a single, variable region that you see here. Similarly, the joining sequences, J lambda 1 means the joining sequence, segment number 1 of the lambda light chain. One segment is here, segment is here, the other segments are here. So all these segments should be joined together in order to make a single joining region. And then in the next step, the constant regions, also there could be a number of segments that code for gene segments that code for the constant part of this antibody, the blue one that you see here, this segment here and here in the lambda light chain. So these should also be brought together in order to make a single, constant region that you see here. So for simplicity, these in the germline label, here in this image, you can see variable joining and constant region of the light chains are given here. So if, this means that this variable region could be a number up to 30 different segments that are present there in the germline DNA. And through the process of somatic recombination, these have to be brought together in this way that the variable sequences and joining sequences are brought together in the first place. They would be brought together, and these variable segments, joining segments, and the constant segments, they would be rearranged so that when transcription of this, would take place, you would find a transcript that would give you a different types of peptide or in each case the constant region actually as the name indicates, the antigen binds here to the variable region of the antibody, this top. So variation is ensured right here when this recombination is taking place and the variable segments are brought together. As I told you, I'm telling you again that the variation, they are brought together in different ways. There are different rules, and then when they are brought together, each immunoglobulin or antibody expressed on the surface of the B cell would, be different from in terms of this variable region of the light chain because of this genetic rearrangement that is happening here. And what are the enzymes that are involved in this particular process? We call these enzymes as RAG1 and 2. These are the main enzymes, although there could be a number of other factors that are involved in this process. But the major enzymes are RAG1 and RAG2. These are recombination activating genes. RAG is for abbreviation of this recombination activating genes. And these genes actually are particularly RAG1. This is very actively involved in this particular process. And this how this DNA cut and paste actually is taking place, in how, where to cut, how this particular enzyme knows where should actually I cut the DNA. So this enzyme is going to cut the DNA at very specific sites, you know. These are called, the spacer sites. And this enzyme has to go there and cut the DNA at the spacer sites, the white sites like this 23 base pair spacer, or here you can say the 12 base pair spacer, and you see some of the sequences. You suppose these are the DNA sequences. This is the DNA sequence in orange color, 7 base pair. This is a 7 base pair sequence along this line. So this enzyme is going to recognize these 7 base pair sequence, or this 9 base pair sequence. This is called nanomer or heptamer 7 base pair sequences. These are recombination signal sequences, or RSS. Okay, so these recombination signal sequences signal these enzymes to go there and bind to these sequences, and then cut the DNA, at the intervening spaces, or the spacer sequences that you see here. So the enzymes are signaled by these 9 base pair or 7 base pair sequences. Enzyme goes there, it binds there to these sequences, and then cut, make a double-stranded break into the DNA strand, and in this way, these segments are brought together. The intervening sequences are removed, and they would just do the rearrangement of the gene. In case of the kappa light chain, if it is a kappa type, again you can see these recombination signal sequences in color, and the white spaces in between them. In the case of the heavy chain, also you see the same type of arrangement, and these RAG1 and 2 enzymes are primarily involved in this particular process. Here you can see that if it is a copper light chain, again, you can find that 35 different variable segments have to be combined to make one variable region of the copper light chain. Similarly, for the heavy chain locus, you would have to recombine about 40 different variable segments found on chromosome number 14. They should be brought together to make this region, this one and this one, which are the heavy chain variable regions. So this kind of recombination in case of the light chain is called VJ recombination. In case of the heavy chain, the variable and diversity D and J and constant region C sequences should be brought together. This is called VDJ recombination, and this is called VJ recombination for the light chain, VDJ recombination for the heavy chain. So this kind of recombination process or gene rearrangement process happens in each and every B cell, as I told you, with the help of RAG1 and 2 enzymes. When the B cell, inside the lymph nodes, it has come in contact with some antigens, then what would happen to the B cell in that case? The B cell would proliferate, its clonal expansion would take place because it has found a perfect partner or an antigen that it can bind with. And now, though that particular cell, B cell, is required, not other B cells are required. So that particular cells, the clones of that cells are made there in the lymphatic system, and those cells would stay inside the cell. Then after this particular process which happens, this antigen antibody matching that happens there in the lymphatic system, and proliferation of the B cells occur. And as you know, that initially the immunoglobulin that is expressed on the surface, this is IgM, right? So Now, the B cell is going to refine this particular antibody after some time. The B cell that has become an effector cell, it would further refine that antibody. The initial immunoglobulin, IgM, which is a pentamer, and it's not a high affinity antibody for the antigen. That antibody should be fine-tuned, it should be refined. Uh, and this process of refinement actually is accomplished with the help of single nucleotide substitutions and our point mutations that are almost randomly incorporated in the antibody encoding genes right here during this particular process that I told you, the genetic rearrangement that was happening before. Now, in the variable regions, these variable regions are important for antigen binding, as I told you. Some point mutation or single nucleotide substitutions would be done, and those substitutions would strengthen the binding power of the antibody. It would increase the binding strength of the antibody when it attaches to the antigen, to the antigens. So, as I told you, IgM is a pentamer, but its binding strength is weaker as compared to IgG, which is the next antibody. It is secreted as a monomer. It has a hinge region. It is flexible, and its binding strength as compared to IgM is more. So, how because in the case of IgM, if you look at the sequence of the antibody in different cases and compare them, only couple of complementarity, determining regions in some cases would have some mutations. These red boxes here, for instance, are showing you different mutations in some of the antibody antibodies, which involves in some cases the CDR2, the complementarity determining two regions in the variable region of the heavy chain, or CDR3 of the complementarity determining regions of the variable region. In the light chain, also you would see very few cases where you can find some mutations here. For instance, here, in CDR2 and 3, and other isolates, as you see there, these parallel lines of different isolates that when you do immunization or when there is some kind of primary infection and you check the DNA, sequences coding for these variable regions in case of the very light chain and heavy chain, you see that very fewer mutations or few mutations are found in the case of IgM, the initial antibody secreted by the B cell, the plasma B cell. But after 2 weeks of immunization or 2 weeks of infections, you would find that IgG would have started appearing in the sera or the serum of a particular person who was infected or immunized. In that case, when you sequence these variable regions of the light chain and heavy chains, so you would find many different mutations that have happened here. The point mutations that we are talking about here, it introduces further diversity into, you can say, in the sequence of the variable region. And that diversity is important for increasing the binding strength of the antibody. It means the B cell now has come in contact with the antigen. It knows that what kind of antibody should be synthesized, and now this antibody is further um, strengthened, this binding is strengthened with the help of this particular, process. And this is accomplished with the help of cytidine deaminase, which is an enzyme AIT, expressed only in the proliferating B cells. So this was about the antibodies. You can see that these antibody coding genes on different chromosomes for the kappa, lambda light chains or the heavy chains, which is on chromosome 14. Okay, thanks.